there, we're almost done. We are Team PANCREAS, which stands for Preventing Adverse Neurological Consequences of Reduced Episodic Amounts of Sugar. My name is Eileen Sayre, and presenting with me tonight are Danielle Wilkin, Lynn Hunchu, and Joe Franklin. And the rest of our team is here as well, comprised of Kelly Gunther, Alexis Matthews, Siddharth Plackett, Melanie Shapiro, Andrew Naran-Shaw, and Peter Wynn. Type 1 diabetes is a common autoimmune disorder found in every 1 in every 400 children and adolescents. And it's where the body destroys the pancreatic beta cells. And the beta cells are the cells that produce insulin for the body. And so insulin is a powerful hormone that is critical in regulating blood sugar levels. And so the body has very complex feedback loops and mechanisms in order to properly maintain a narrow range of blood sugar levels for homeostasis. However, when the body cannot produce insulin, these feedback loops are disrupted. And so patients with type 1 diabetes must supplement themselves with insulin through the use of intensive insulin therapy. However, this presents a lot of challenges because it's very difficult to approximate the amount of insulin you need to reach your target blood glucose levels. Your body does an amazing job of it on its own, and so it's very difficult for us to do it externally. And so a risk associated with intensive insulin therapy is hypoglycemia, or low blood sugar. And this is dangerous because in your brain, you have a very high metabolic demand for sugar, and in hypoglycemia, these demands are not met. And so in acute episodes of hypoglycemia, certain side effects include impaired behavior and judgment, but in severe cases, permanent brain damage can result with further decline in behavior. And so a way to combat this is through the use of preconditioning, which can broadly be defined as exposing an organism to a stimulus in order to prepare that organism for exposure to the same stimulus in the future. And so a common example of this is through the use of a vaccine, where an organism is presented with a weakened version of an infectious agent in order to prepare it for the future for exposure to the same infectious agent when we fly. And so we adapt this definition of preconditioning for our project by defining it as inducing several moderate episodes of hypoglycemia before a severe episode. And so this has been known to ameliorate some of the negative effects observed in hypoglycemia and lessen the damage, mostly seen through necrosis in the brain and behavioral deficits. And so within the brain, we are primarily interested in looking at the hippocampus, which is responsible for mediating learning, memory, anxiety, and spatial awareness. Specifically, we're looking at the dentate gyrus, which is a sublayer within the hippocampus and is representative of the hippocampus itself. It also displays a high rate of neurogenesis, or cell birth. And so increased death within this part of the brain is linked with some of the behavioral deficits and cognitive decline. And so a mechanism to sort of explain preconditioning biochemically is through the use of lactate as an alternative source of fuel. And so during hypoglycemia, neurons are able to use lactate when blood glucose is not available. And so this works because lactate is able to be converted back into pyruvate and then used as a substrate in the citric acid cycle in order to produce ATP. And so lactate infusion has been shown to decrease some of the neuronal death associated with hypoglycemia by approximately 80%. And so lactate moves around the brain through the use of monocarboxylate transporters, or MCT proteins. And these are integral membrane proteins that shuttle lactate in and out of the cell. And so MCT proteins are upregulated in the brain in two types of cells during hypoglycemia. So on the right is an astrocyte, which is a cell that is used to metabolically support the brain and stores lactate. And so during hypoglycemia, there is an increased amount of lactate, and in order to shuttle this increased amount of lactate, there is an upregulation of MCT1 proteins within the membrane of the astrocyte. And so this is to transport the increased amount of lactate out of the cell and into the cell on the left, which is a neuron. And the neuron is the cell that needs the energy because there's not enough glucose, and so it will upregulate its production of MCT2 proteins in order to uptake the increased amount of lactate for the brain. And so our research questions are as follows. Number one, how does preconditioning severe hypoglycemic episodes with multiple moderate episodes alter behavioral performance? Number two, how does preconditioning alter neuronal death caused by severe hypoglycemia? And number three, how does recurrent moderate hypoglycemia alter the expression of the lactate transport proteins MCT1 and MCT2 within the brain?
So as Eileen said, we are going to be inducing hypoglycemia, and we're going to be using a rat model in our experiment. Obviously, a very important thing is determining what blood glucose levels constitute a moderate versus severe episode of hypoglycemia. In our first experiment, we will be inducing moderate episodes of hypoglycemia, followed by a severe episode, and in our second experiment, we will be only inducing moderate episodes. So for our moderate episodes, as you can see, we're shooting for a blood glucose level of approximately 25 to 40 milligrams per deciliter. This will be maintained for 3 hours, and then terminated with an dextrose injection. Our severe episodes will have a much lower target glucose level of 10 to 15 milligrams per deciliter and be maintained for about 90 minutes, and they will also be terminated by injection of dextrose. Throughout these episodes, we will be extracting blood from the tail vein of our rats and using a freestyle glucose test meter to monitor the blood glucose levels. So our first experiment is attempting to answer the first and second research questions that Eileen talked about earlier, specifically the behavioral consequences of preconditioning as well as what it does to neural death. So our first behavioral test is the novel object recognition test, which assesses recognition memory. As you can see, we will place our rats in the first box in sample 1, which will have 2 identical objects. Our rats will be given 4 minutes to explore and then removed from the box for a period of 1 hour. They'll then be placed back in the box with a separate set of identical objects and given another 4 minutes to explore. Following a 3-hour break, they will be placed back in the box with 1 object from sample 1 and 1 object from sample 2, and their movement will be tracked using an ethovision camera. In order to assess recognition memory, we will be measuring the amount of time spent exploring each of the objects. And typically, rats who spend more time exploring a more novel object, which in this case is the object from sample 1, will have a more intact recognition memory. Our elevated plus maze is our second behavioral test. It consists of 2 open arms and 2 closed arms. The rats will be placed in the middle for a period of 5 minutes and given time to explore, and their movements will be tracked using the ethovision camera used in our novel object recognition test. This test is used to assess anxiety-like behavior, and typically rats who spend more time in the open arms have lower anxiety-like behavior than those who spend more time in closed arms. And finally, we will be quantifying neuronal death using a fluoride B stain. As Eileen said earlier, we're going to be examining the dentate gyrus. This fluoride B stain fluoresces green using fluorescence microscopy, and it has a high affinity for necrotic neurons. So we'll use this to determine the necrosis caused by our severe episode of hypoglycemia. So for our timeline, we will receive our rats, and they will have a period of habituation to get used to their new environment. We will have 3 experimental groups, one which will receive no hypoglycemia whatsoever, be that moderate or severe. Our control group will receive saline injections instead of the insulin injections for the moderate episodes, and then they will receive the severe episode of hypoglycemia on the 4th day. And our experimental group will receive insulin injections to induce moderate hypoglycemia on days 1 through 3, and then have the severe episode of hypoglycemia on the 4th day. This will be followed by a period of rest, after which we will begin our behavioral experiments. First, our novel object recognition test on day 11, then our elevated plus maze on day 12, and then afterwards we'll perfuse our rats and then perform our fluoride B stain. So for our expected behavior results, we expect that our preconditioned rats will spend more time with the novel object in the novel object recognition test. This will show that they have an increased recognition memory as compared to their non-preconditioned counterparts. And for the elevator plus maze, we expect that our rats who have been preconditioned will spend more time in the open arms, indicative of lower anxiety-like behavior. And for our FJP, we believe that our preconditioned rats will have a lower number of necrotic neurons as compared to the control groups which did not receive preconditioning, which demonstrates the neuroprotective effects of preconditioning. And now I'll pass it on to Lena.
So in experiment 2, we are attempting to answer the 3rd research question, how recurrent moderate episodes of hypoglycemia affect MCT expression in the brain. So in order to do this, we have proposed an experimental timeline in which rats will be assigned to either the control group or the experimental group. The rats will then be removed from the testing group at 3 distinct time points following either saline or insulin injection. We will then perfuse the rats and perform immunohistochemistry. So in order to quantify MCT1 and MCT2 protein expression, we'll be using a double immunofluorescence staining technique. In this image, panel D and E depict cortical neurons stained with GFAP, an astrocytic marker, and MCT1 in panel D and MCT2 in panel E. Fluorescence will then be quantified using epifluorescence microscopy. So in experiment 2, we expect to see a general increase in MCT expression as we increase the number of moderate episodes of hypoglycemia. And now to summarize our expected results, in experiment 1, we hope to confirm that preconditioned rats will maintain recognition memory and exhibit decreased anxiety-like behavior, as well as decreased neuronal death compared to the control group. In experiment 2, we hope to see an increase in MCT expression with increasing number of hypoglycemic episodes, which will suggest a neuronal adaptation for alternative metabolite usage. And now I'll pass it off to Joey.
Our Gemstone program timeline is as follows. In spring 2013, we received the Howard Hughes Medical Institute grant, which is helping fund our research. We also presented to our thesis proposal to a committee of experts and received their feedback. In the summer of 2013, we obtained the Institutional Animal Care and Use Committee approval, which is a committee that is needed in order to use animals in your experiment. In the fall of 2013, we are conducting a pilot study. We've also written chapters 1 and 2 of our thesis, and I am presenting at Junior Colloquium. In the spring of 2014, we are going to conduct experiment 1. We will begin analysis of our data, finalize the first 3 chapters of our thesis, and identify the experts that we will be presenting to at our senior thesis defense. In the fall of 2014, we will conduct experiment 2, complete our data analysis, revise our thesis draft, prepare our presentation, and submit the names of our experts, which we will be presenting to at our senior thesis defense. In the spring of 2015, we will complete our senior thesis. We will have our senior thesis defense, and please God have our citation ceremony. Our present status is that we have completed all laboratory training, we have outlined our thesis, we have written drafts of chapters 1 and 2 of our thesis, and we have submitted a protocol amendment to ICOOK in order to properly assess the correct dosage of insulin, and then we were going to use that to just change a little bit of our experiment 1. Challenges that have faced us that hopefully won't face you now that we're telling you about them. Not obtaining ICOOK approval the first time. ICOOK is a tedious process that requires multiple submissions, so make sure to allocate a lot of time because you probably will have to submit a lot of time, so just make sure you have that built in. Another challenge would be completing all on-campus lab trainings. This one seems pretty straightforward, but there actually is a in-class training component of a lot of these trainings, which you have to fit into your schedule. So make sure you also allocate time to fit that into your schedule so that it doesn't hold you up from starting your experiment on time. Another challenge that I alluded to earlier is allocating plenty of time to correct unforeseen challenges to your experimental design. We actually had a challenge to our experiment 1 where we didn't really know the correct dosage of insulin to induce hypoglycemia. Therefore, we have to do a pilot study this semester. So just make sure to give yourself enough time and enough cushion to really build in time in case there are these challenges. This is pretty straightforward one. Coordinating team meeting times. We're all very busy taking a lot of different classes, so you might have to have some 8 a.m.s, unfortunately, but it'll be all right. Finally, team dynamics. You've heard a lot about this, and I want to stress this to you. It's probably really important, is that you really have communication. Gemstone is built on teamwork, and in order to be successful, you really need to have good teamwork. I know that's really simple, but it's really important. And finally, you should always make sure to utilize your mentor's expertise. She, in our case, is a she, but in your case, it could be he or she. And they're there for a reason, and so make sure to lean on them, and because they will definitely help you out. At this time, I'd like to thank our mentor, Dr. Glasper, all of the graduate students, lab assistants who have helped us in our research, the Gemstone staff, our librarian, Dr. Bekucheva, and the Howard Hughes Medical Institute for funding our research. And at this time, I'd like to thank you all for listening to us and looking forward to questions.